Cascades of similar signaling events are a common theme in biology. We see kinase cascades throughout eukaryotic regulation, and it is also common to see transcription factors controlled by other transcription factors. When I was in school, it was not understood why these cascades occurred. It was falsely explained as some amplification of signal behavior. It turns out that these cascades do something more subtle to a signal, and that's what's being explored in this study. In this study by Weiss and co-workers, they explore computationally and experimentally what happens when you daisy chain several transcriptional repression circuits in a row. Let's start with the wet lab experiment. They describe the same repressor proteins, TEDR, LAC-I, and C1, as we described with the toggle switch and repressilator, but instead of being arranged cyclically, these are the open designs. In each circuit, the cells are turned on by addition of ATC, which will derepress the TET promoter at time 0. That initiates the production of the first protein, which is LAC-I in all cases. Circuit 1 is the simple case of constitutive expression we described in an earlier video. In circuit 2, we have an intermediary inverter, and in circuit 3, we have two such inverters. In figure A, they describe the fluorescence of the cells as a function of ATC concentration. So in effect, they are tuning the knob of input promoter strength and monitoring the output of each of the three circuits. You'll notice that with no intermediary inverter, the system is monotonically increasing with signal. With the single LAC-I inverter added, the signal is inverted, and with the second inverter added to the system, it's monotonically increasing again. They also note that the slope of the response is greater with more stages. Mathematically, this can be shown to require cooperativity of repressor binding. If the repressors are monomers, this ultrasensitivity effect is not observed. In figure B, they plot the variability from cell to cell as a function of fluorescence. They find that with increased numbers of stages, the variability increases. This is postulated to be the result of noise amplification. They also observe that the response time becomes more heterogeneous as the number of stages increases. All these behaviors can similarly be captured in the differential equation and stochastic models they do, and it correlates well with the experimentally observed values.